Welcome, everyone, to the third webinar of the series on implementing targeted next-generation sequencing for detection of drug-resistant TB in high-burden countries, from recommendations to reality. This series is being organized by the Stop2B Partnership and FIND in collaboration with USAID, UNITAID, the Global Laboratory Initiative, and the New Diagnostics Working Group. My name is Wayne Van Gemmerts. I'm a Diagnostics Technical Officer at the Stop2B Partnership.
And I'm Anita Suresh, Head of the Genomics and Sequencing Unit at FIND. We're really happy to have you here for this webinar that we've developed, really specifically targeting the TB-affected communities in civil society. The first two webinars, in case some of you all joined that, were more technical, and we know the audience has more been focused on lab scientists and researchers and so on, and policymakers. So I think with this webinar, we want to really get into the basics of, some basic understanding of how sequencing works, and more importantly, its potential for rapid identification of TB drug resistance, and also the why, why should we care about this tool and how can we make this actually useful and help the TB community with this tool. So we really hope this will provide a foundation for advocacy for the uptake of sequencing for drug resistance testing, especially alongside the rollout of the new regimens, because that's especially specific or big role that sequencing can play alongside the rollout of the new short regimens and all the other newer drugs and repurposed drugs. And so, yeah, that's where I'm really excited about going ahead with this one.
Okay, a couple of logistics details. This webinar is being recorded. We'll share a link afterwards, as well as the presentations. And if you haven't seen it yet, there is an option for simultaneous interpretation in both French and Russian. So if you click the More button at the bottom of the screen, then you click Interpretation. There are going to be two moderated Q&A sessions, and we encourage you to take advantage of the Q&A box to write your questions. You will also have the option to raise your hand to be unmuted in case you want to verbally ask your question. We're happy to have with us today two TB advocates who will be moderators for this sessions. We have Sarab Rania and Goodman Makanda. But first, I would like to introduce our Executive Director at the Stop2B Partnership, Dr. Luchika Ditu, who will share with us some welcome remarks for the webinar.
Thank you. And so I want to thank very much you, Anita, and obviously you, Wayne, our colleagues from USAID and the teams for making this happen. And I want to thank the 140 people that are online today. It's very encouraging to see this large number. It's also a dilemma that I have, and you all know me, that we have 700 people that registered for it. And we have 150 that joined. I think that will be the number. So I always wonder why people register in such a large number if they don't want to attend, but that's a different conversation. 150 is a very, very good number to have. And it's great to see this interest, especially because we have those of you here representing civil society, communities, TB survivors, in a webinar that I think is a great idea, because very often, and I want to confess that even for me, when you look at the tools, the new tools, all new tools, including treatment regimens, including treatment drugs, including new tests. Sometimes we speak about them, and very often we don't really understand all the things that are meant. So I'm really, really happy that we have with us as well a lot of our challenge facility for civil society partners and grantees to learn about sequencing, to learn the basics, what is it, and to know why and how we can advocate for uptake of these tools in the TB high burden country. And obviously, having so much of actually all these tools are developed to be used, okay? And if they are not used, and they are not used for diagnosing people, for getting better access to treatment and increase the survival and the quality of life, all these tools are useless. So that's why it's extremely important for us to understand all these pieces there. I want to recognize as well our Vice Chair of the Board, Mr. Austin, here with us. Thank you, Austin, for taking the time and for the leadership that you do all the time in pushing this forward. Now, we know very well USAID as well as l'Initiative powered by Expertise France are the ones funding Challenge Facility and therefore engagement of some of you in these efforts. Now, I want to make 3 points. Actually 4, because there is a last one which I think is very important. First one is that the UNHLM in 2023 calls for around 1.5 million people to be treated, diagnosed and treated for drug-resistant TB by 2027. I want to tell you that a few months ago, and Wayne knows, I was very discouraged because I realized that in terms of drug-resistant TB, we are kind of going in circles, guys. In 2022, less than 180,000, basically around 176,000 people were enrolled on MDR or resistance treatments. And this number is lower than the one in 2019, but it's also, you know, when I was younger, several years ago, we were speaking about 250,000 to be diagnosed and treated in a year, and we had that target. And I think me and Wayne, who know each other since we were in Copenhagen discussing this in WHO Europe. And it's a bit that we are below 200,000 in the year 2024. So the first thing is we really need to move in a smart way about diagnosing and treated people with drug-resistant TB properly, starting now, I would say. Now, the second point is that we have 2 new drugs. We cried all the time for new drugs. There is so much demand. So now we have the BPAL, we have a BPAL regimen that includes the 2 new drugs, which are the bedaquiline and the pretomanid. Obviously, the repurposed drug linezolid, but as you know, we don't have the tools, the automated tools to do the line probe assays to test for the resistance to these drugs. And the phenotypic culture and DST can take a long time. So we know that there is a sort of blinding treating people. And also, we are at the risk of losing these drugs, which will be really bad if we are provide this regimen to people who have resistance to 1 or more of these drugs. So this is nothing new. And I know you guys, you will refer probably to it on the fact that we observe already resistance to bedaquiline. And sometimes, you know, this is a talking point. I'm saying it goes. Let it sink. If we lose bedaquiline, if we lose pretomanid, we are doomed. Okay, doomed, really. It's not an exaggeration. It's not just to scare people. So we need to handle it with care, I'm trying to say. I don't want to go into the in the past eras where we had to greenlight everything and was complicated. People need to have access, but we need to be very careful to whom we give it, how we monitor in such a way that we don't lose these drugs. Now, the sequencing can detect resistance for bedaquiline and the linezolid rapidly for other drugs as well. I know is not the best, Wayne says is not the silver bullet. You will hear why not. But it is a very powerful tool for drug-resistant TB diagnosis. And obviously, it will become more and more relevant and powerful as the science advances. So it's very important for everybody here to understand what the targeted next generation sequencing means. It's a long name even to say it all. Last night, I was speaking with somebody who was not in health and for sure not in TB, and said, that's such a complicated title, what it means. So and he said, okay, well, call it something easier. But anyway, you will hear about it. And I know everybody here is much more proficient than me and my friends. And I hope that with this, the TB community understands one piece of innovation and will embrace it and push for this to be rolled out. And I know that a lot of work supported by Global Fund, by WHO, by countries and other implementing partners is taking place. My closing point has 3 subpoints. One is, very good we do this webinar. We should do for every single tool. We should do for every other drug. People need to understand what these are, why they are important, why are we in need to scale them up and to use them, and what's the benefit. Number 2, next week, in 2 weeks is the UNHLM on AMR. Drug-resistant TB is part of the antimicrobial resistance. We have a declaration that is very strong, very powerful, and include on antimicrobial resistance that includes reference to TB, including to Stop TB, including to the work of GDF and other teams, which is really good. It should not be about that. It should be about making sure that TB and AMR are seen together and that TB is seen as part of the AMR. And I want to thank all of you for making sure that this is the language in that declaration. But it's very important to make sure that we are continuing on this and that we are highlighting not only in New York, but in all our work, the links between this. And also I'm happy that Austin, as I said, is here to speak about the key asks of the TB community on the UNHLM on AMR. The last point I want to make this, but nothing of this can happen if we don't have the money, okay? And in TB, not in TB diagnosis, in general, we have a huge gap in terms of resources. There is an entire agitation that will happen exactly this month and the other one before the Global Fund Board to request that the TB share among the Global Fund funded diseases, HIV, malaria, and TB is increased. For too long, TB got just 18% of the funding, even though TB kills more than HIV and malaria. TB gets less of all. 50% of Global Fund money go to HIV, 32% go to malaria, and 18% only to TB. None of this discussion today will have any relevance if we don't have money. At the current level of funding from Global Fund, we are not even able to cover the diagnosis using the gene expert and so on. The sequencing will seem like me telling you, and tomorrow I go on the moon. You'll be like, yeah, you want to, but you will not, okay? So why I'm saying this is this, and Austin, maybe you add one point in your speech. There is a petition going on that the world that cares about TB needs to sign. So I make a plea to 180 people here as individuals to sign this petition. I make a plea to Austin or the colleagues here. Is not a Stop TB petition, is a TB community petition demanding more funding for TB, because all these conversations will remain really good, really educational. We will all be smarter, but it will not reach the people that need it. So 181 people, 179 now, 2 people, no, okay, 181. There is somebody who's not decided to stay or go. 181 signatures on the petition. If you care about the people with TB and if you care about people getting access to the tool they deserve, including the sequencing. Wayne and Anita, apologies for taking your a bit more time. Thank you very much.
Thank you so much, Luchika. So let's move on to the first presentation of the webinar, which will be given by Anita. She is going to present on why does sequencing matter for TB care. So over to you, Anita.
Thank you, Wayne. It's a very tough act to follow after going after Luchika anytime, but she highlighted the exact most important ones. So that helps me also to really focus the message of the content here for the sequencing around that. So really the goal here is why does sequencing matter for TB care itself, right? So let's start really with some of the basics. Again, you've heard these different terms, as Luchika said, lots of complex terms, tNGS, targeted sequencing, whole genome sequencing, NGS, like all used, sort of or interchangeably almost, just sequencing or next-gen sequencing or next-gen sequencing. So a little bit on just let's talk about some terms, some basics, so that as we present or as you get to more talks and have these discussions and have those the sort of advocacy conversations or conversations with funders, with the governments, with health programs. It's key that I think having those basics clear would really help you have those and get really to the crux of the matter. So I think we talk about genomics all the time. Genomics is overall, it's just a study of all of the entire genome. So it's really the overall picture, not just so sequencing is a technology. Sequencing is the tool or a part of genomics. So genomics is sort of the study or research of any organism's entire genome, DNA, RNA, all of its genetic material. Sequencing is the process of actually determining that order of those nucleotides or of all those base pairs in the DNA or RNA. Now, next-gen sequencing, and I'll keep, again, we may say sequencing just because almost every sequencing new thing that's coming out, everything that's there is really next-gen sequencing, and this really came out in before 2000. So really right now it is the current. So when we say sequencing, most likely people are meaning next generation sequencing. And the beauty of NGS, the short form here, is essentially it's massively parallel sequencing. So large amounts of DNA and RNA doing the same sequence. I'm going through the AP, GC, all of that, but I can do that multiple millions of them at one time. So the number of copies of that I'm getting hence increases the sensitivity of the test itself, of the result. And what you're going to see is the variance, right? When you're looking at the ATCG, all of the sequence, if there are mutations, if there are variants, basically changes from what it's supposed to be, that most likely means something. And that is where, in this case, in the case of TB, we're talking drug resistance. In the case of many pathogens, it's going to be drug resistance. You know, in the case of COVID, those variants were associated, many of the variants were associated with either more infectivity, more spread, easy to spread, more sort of contagious, a lot of those different aspects. So the variants, mutations really are the ones that have that information, which is what you want to be able to say, this change means something, and I need to do this action based on this change. Of course, most of you would be familiar with PCR, or qPCR, or just PCR, which essentially is the same thing, right? It is talking about sequence, in a way, looking at targets, looking at the DNA. It is also a molecular test. So we are really talking, all of these are molecular tests. Sequencing is also a molecular test. But the big difference is that PCR is looking at it's basically looking at limited genes or even limited regions or specific mutations in a few targets and very known regions that you're looking at in PCR. Whereas with sequencing, in this case, talk about targeted sequencing or whole genome sequencing, we can simultaneously sequence hundreds to thousands of genes at one time. So no matter what the gene region is, no matter how many genes, can all be done in one test. I think that's an important sort of differentiation. So if you know what you're looking for and you're looking for one or two things, PCR is the way to go. But if you're then talking about looking at a whole profile, all of the resistance, all of the differences, all of the things you should act on to make it clinically actionable and actually helpful for the person to say, what should I do with these differences, with these mutations? That's where you go for sequencing. So in this webinar, we won't talk so much about the different products, but you, again, just more to get those terms familiar, you would have seen them around, which is different companies that are out there, Illumina being the largest one in the largest install base in the world today, Oxford Nanopore being also a very close second, with also a lot of growing install base especially in sort of in the low and middle income countries. And then, of course, there's BGI or MGI, which is a sequencing company of products out of China. Thermo Fisher also has sequencing reagents. They were the ones who did the original traditional Sanger sequencing, but they also have newer sequencing equipment these days. So that, in a nutshell, is all of the key terms. Anything I use or anything you henceforth, going forward, go to any sequencing talk, at least you'll know what we are talking about. So a lot of times these will be skipped and we'll just say sequencing or NGS. So we're really talking about the same thing. So now I've said all that. Now let's put it in the context of TB. As Luchika was saying, it is about drug resistance, right? So basically, yes, there's an idea of TB detection, identifying yes or no for TB, potentially using something like an expert or a other molecular test to get a resistance reading as well for a RIF and an INH for one or two of the key drugs. But really for a full profile, a comprehensive profile of all the resistance, especially for the newer drugs, today the standard of care is culture-based DST or drug susceptibility testing. This is what we say phenotypic because it's about what you can see, it's the phenotype versus what you can't see, which is molecular. So this, as you know, takes 6 to 9 weeks. So I don't think we should ever talk about a diagnostic that takes in the matter of weeks, right? This was what the best we had for a long time, though. There was nothing else. This was a good solution for a long time. But we keep evolving as technology evolves. There's more options, and we need to take advantage of that for TB as well. It shouldn't just be basically disease priorities of sort of high-income countries that take advantage of these technologies. So here's where you're seeing then targeted sequencing can pretty much do that comprehensive testing, resistance testing for TB, but in a matter of 2 to 3 days and without having to do culture. So really starting directly from the clinical sample or a sputum sample in this case. So we're really talking about a paradigm shift when it comes to drug-resistant TB diagnosis using something like targeted sequencing compared to the standard of care. So no other test today can actually do a molecular test that can do drug resistance testing for these, especially for the new drugs, the bedaquiline, clofazimine, and linezolid. So I'll talk to you a little bit about what that involves, but I think that is the key for you to look here. What we're talking about is today, the DST, to be able to do DST for all of these drugs, the new drugs, the repurposed drugs, we need to do culture. And that in most cases probably is not being done, or people are being put on treatment before the test results are even there. And then the treatments are adjusted depending on whether it worked, whether it didn't work, and what the results show, which is really not the way to be going. So this is a test here we're talking about that can actually give you that results within the 2 to 3 day time frame to be able to start everyone on the right treatment from the start. So you would say, why isn't this out there already? Well, for a number of reasons. One being, it is still a little complex, as already I spoke about all the different terms, but it is still a workflow, meaning there is still a few parts to getting the test done. So it's not just one box as you have ideally for other things, as you have a PCR test. Today, it still requires 3 components. So it requires the DNA extraction, it requires the library preparation, and it then goes into the sequencer to actually do the sequencing, which is actually the most straightforward part because you have something that you put in that can sequence. And then you do the data analysis to get the result reporting. So all of the products in the market or the products that we've looked at, they actually combine all of these. So they offer all of these. So when you look at getting sequencing, or countries are looking at getting a sequencing setup, it is about getting this entire workflow, including the bioinformatics, so that the result that comes out, you don't need a sophisticated bioinformatics lab to be able to understand the results and interpret and utilize them as a clinician to understand them. So as I said, some of the advantages already, this is again to highlight, as I said, direct from sputum sample, which means you don't have to do culture. The resistance that you can detect for as many drugs as knowledge exists in the world today. And as Ruchika also mentioned, this will only grow with time. We already saw a change, a difference, an increase in the last 2 to 3 years, and this will continue to increase. As long as that's captured, this test can also show that, and so you can continue to get the resistance captured in these tests. Faster turnaround time I mentioned. The other aspect, of course, is not the need, basically removes the need for a BSL-3 or the biosafety level of 3 that is now needed for culture, because obviously with culture, you have you're growing up bacteria to very, very high concentration. Here, you're pretty much extracting DNA, then you're not having to worry about the live bug itself. And the last one I would highlight is that it is high throughput. So again, that is something it's come depending, when I say high throughput, it also means it's flexible. So depending on the need, depending on a setting, depending on a district or a city or a higher concentration central area, you can have as much throughput as you need. And that is the sequencer that setting should have. So you do have the flexibility. You can maybe doing 10 samples in a week, or it might be processing 100 samples in a week. So for each of that, there is different throughputs. You can, of course, scale, go from small to big, or add more and so on. So that really helps you in terms of how long it will take. So the more you can put on a sequencer, you can get one run, get the results out, move to the next batch. This is just a plug for the project that we did. So now, yes, we know there's technologies out there, and we know that, there's potentially they can work, they can have a potential. But I think, so this was 4 years ago, it was really about, but where is the evidence, or is there a standardized way we can actually evaluate these products to know how they work and how they make sense, especially in settings of intended use. So a lot of these products were made at different points in time, but really not utilized. They're used for research studies, various maybe surveillance projects, but not really used in a very controlled clinical setting in a large scale to understand how does it work with clinical samples from the field. So Seek and Treat was very much about, which was a grant we got, which is funded by Unitate, which FIND has been working on with several partners, was essentially to generate that evidence and to also catalyze the in-country capacity for sequencing, to be able to support the adoption and rollout of targeted sequencing, targeted NGS, for the comprehensive diagnosis of DRTB. I'll only highlight a couple of things here. Is one, the clinical evaluation that was completed last year, over a year ago now, was done in India, Georgia, and South Africa, really representing as diverse as possible in terms of health systems, sort of health system settings, but also prevalence, also the DRTB incidence, all of those, the data, slightly different varying epidemiologies in these countries, but also health systems wise, how sequencing would work here. Now, these results were then put together, shared with the WHO. And earlier this year, you'd have seen, we're very happy to see that WHO updated their policy guidelines to recommend the use of targeted sequencing for drug-resistant TB diagnosis. So this was obviously a very monumental step. It was a brand new class of technology, as you saw. It's very different from the PCR, although it's still molecular. It's still a bit more to think about. The fact that you can get so much data, so many answers, is still something to see. Can it actually then, how will this be utilized, knowing what health systems already, all the limitations, all the existing concerns, and also the limited budget that's there. So that still is there, but I think the fact that it was looked at saying, given there is no other test, no other molecular test, rapid test in a way for the new drugs, it was clearly seen that sequencing today and can continue to be the diagnostic that can actually support as many drugs to give you the resistance profile, but also as with changes in drug resistance as it evolves and emerges in the world. One of the things we mentioned, of course, is cost. Now, this is still ongoing work. There is a costing analysis that still needs to be completed. There was a bit of this done. It's obviously, to some extent, there is, you can imagine if there's 3 boxes, there is going to be an expense. It is going to be more expensive than buying a PCR machine. So there is a capital investment needed to get to sequencing, but it's about how effective is that? What does it mean in terms of how much can you get out of it? And what does it mean for overall for the health program, for the TB program, in terms of what it means in terms of eventually translating to hopefully more diagnosis, and hence eventually more put on the right treatment from the start, and hence less AMR, but also more lives saved. So these are work that's ongoing. One of the things we're doing is implementation pilots that we are supporting, that countries are leading, that FIND and partners are supporting in Brazil, Indonesia, and South Africa. These are all things that I can happy to talk more, maybe in the questions, or if there's follow-ups needed, this is something we can take on, but I wanted to give you a snapshot of all the pieces of work that have been done and are ongoing as part of this grant, because each of this touches in terms of where and how this technology can be utilized broadly for the program, for programmatic use. So I mentioned the policy. Again, this is something, full policy is published. It's of course 100 plus pages. So you may or may not have read the whole thing. What I want to highlight is that it's essentially talks about really the recommending the use of sequencing. And the idea is that it looks at both situations of people with bacteriology confirmed pulmonary TB, but also for people that have already a RIF resistance, already have an RR or an RR and an INH and RIF resistance pulmonary TB. Now, in those cases, obviously, you want the rest of the results to be based on your treatment or based on sort of the algorithm that the country is following, based on the treatment regimens. So this is where sequencing then has a clear role, where it can really give you the answers to all of the drug resistance, including RIF and INH, but also all the new drugs, as I mentioned. There are 3 products that were evaluated, whose data was evaluated by WHO. One being the T-Plex from GenoScreen with Illumina. The second being the Xampor TB from Oxford Nanopore, and the third being TB-Seq from Hangzhou, from Shengting, from China. Now, obviously, each of these products are also looking, each of these manufacturers are looking to saying, okay, this was great, they're in a way recommended, but they're recommended based on different drugs. So based on how the results looked based on the sensitivity for different drugs. This is the one thing we said with increasing knowledge and hence translating the knowledge, this can improve over time. So each of them obviously is looking to see what is needed to do further improvements so that then this can be again when the products can be looked at again when there's fresh evidence to look at, can we even make them even with the set performance even better? Can they look at more drugs? Can they detect resistance to more drugs? But this is more to say this was an overall view of where the policy stands. So that was one of the big goals of this. But of course, it doesn't stop there, right? Again, policy is all very great. Again, as Lichika was saying, that's great. That's great on paper. We have this. We can say we checked the box. But really, truly, we have to make sure this actually makes a difference in the world. And so we know it can. I think now we know based on results, based on data, that it can result in faster and more informed treatment decisions. So really being enabling people to get started on the right treatment right from the beginning, so you're not sort of messing around or kind of questioning or going back and so on, which unfortunately we know has happened in the past or is happening in places. But really to reduce that risk of AMR, be it acquired or transmitted and so on, you want to reduce that risk. So this is something that can truly help with that. Obviously, surveillance is a big part for TB management. So in terms of understanding where the resistance is emerging. So it need not be about individuals affected with TB, but really about overall, when you look at that and you get the data out of it, statistically looking at enough samples to say, when you do the DR surveillance, what are you seeing emerging? Are you seeing more resistance for RIF, or is more within RIF resistant? Is there more resistance to fluoroquinolones or to bedaquiline? Are you seeing that increase in certain countries where treatment has been running for earlier than other countries, and so on? So you know, with AMR, TB, or any other pathogen, resistance is inevitable, unfortunately. Bugs are going to get resistant. It's about how long you can stave that off. And as Jika, again, she used one of the points that we always use is also to protect the drugs. So definitely protecting the people, but protecting the drugs is the most important thing that we want to do, right? Especially because now we finally have the new drugs that have shown to work so well, these short regimens that are shown to work so well. That's crucial that as many people can benefit from these regimens for as long as possible. So that's where there's more to be said here, but essentially to say that the technology itself allows for then these updates. So as we do surveillance, we understand maybe there's new emerging resistance. Maybe we're seeing some mutations come about that we're like, hey, we haven't seen these before, but there's about 5 of these that I know from based on sort of treatment or how they responded to treatment, there is resistance to this and this drug. This is data then is crucial to feedback that will go to feed into the things like catalogs, but really to feed into these diagnostic tests, sequencing-based diagnostic tests, make those updates so that these tests now can also detect those right from the start. So really a way to keep up with emerging resistance, sequencing is a technology that allows you to actually keep up as close as you can to make sure you're not always late to the boat, essentially, to find the resistance to do something about it. And I want to close with this one thing. One of the things, of course, there's limited funding, all of that, but we do know what happened during COVID. As much as it's a few years ago and we want to get out of that narrative, I think it's important to know that there was so much NGS capacity built around the world. So this is just WHO data from last year, and this has actually gone up even more, but there was a 40% increase in NGS capacity even in 2 years compared to 2021 or in 2022 itself. So we saw that increase in a very short period of time. And you saw that for diagnostics, you saw that for uptake. Now there was disparity, of course, in terms of how much tests reached, how much vaccines reached and so on. So that continues to be the good fight we need to be fighting in terms of access and basically reducing those disparities. But the most important thing here is that there is that capacity that was built for the pandemic, that countries now have, that it's part of the ministries of health, it's part of national reference labs. Find and others also did a mapping. So that's something we've been tracking over time. We continue to track in terms of the NGS facilities in countries and where they are located. And really, they're located in places where should and can be utilized for everything else, not just COVID or influenza. And Global Fund as well. I mean, they have funded, but they funded sequencing heavily for genomic surveillance during COVID. It was part of their larger C19 RM funding, of course. But very clearly also made the case point that this should now be utilized for all of the other, their standard ones that they focus on, which is TB, malaria, and HIV. So this is where, again, there's a real opportunity, and some countries have already taken advantage of this, put this into their global fund request, saying, we have these sequencers in country, but we want to be able to use it for TB. What that would mean is that I can use the same system, I can use many of the same reagents, I can use many of the same consumables. What I would need is a TB-specific assay, for example, the GenoScreen one or the ONT one. So that's a kit or a product. Otherwise, everything else, and that comes with the kit comes with the analysis. So those two are the ones that make it specific for TB. Otherwise, everything else is sitting there. You have people that are trained on sequencing. If they're trained to do sequencing on one pathogen, it is a matter of sort of getting those 2-day, 3-day training on another pathogen, if this is something they have the access to those samples in these settings. So I really want to emphasize this last part, other than the importance, if we said, of overall for NGS, the fact that it can make that difference. The goal of it for diagnosis is protecting the people, but also protecting the drugs so that they continue to remain efficacious in the long term, and we continue to get as much as we can from these really, really good drugs that we have after decades. But the other point is also that it is critical to utilize this NGS infrastructure. We are not starting from scratch. We should not be made to feel that TB is starting from scratch. We have that infrastructure and capacity that was built for the pandemic. And why shouldn't TB be able to take advantage of that? Why shouldn't TB get the benefit of all of this that's sitting there, the infrastructure that's sitting there? So I think those two are the big points I wanted to highlight here. Happy to get into any more details you want, but with that, I'll stop here. And thank you, everyone, for your time.
Thanks, Anita. So we're going to now pass the microphone to the moderator for the Q&A session, Saurabh Rania. Saurabh, the floor is yours. Please be sure to introduce yourself.
Sure. Hi, everyone. This is Saurabh here. I am a TB activist and advocate, and I make sure that I bring all the voices from the ground of the patients that we are actually helping, all of us together, to all the forums like these. And just want to thank Anita. Thank you so much for breaking this down and making it super easy for everyone to understand. As you know me, I would first start with the patient. So this looks like it's a very promising thing to have in the field right now, but what would it take to bring it to scale? So my question would be, what are 2 or 3 of the biggest challenges to bring this to scale in low and middle income countries?
Thanks, Saurabh. Yeah, and I always can rely on you to bring the hard questions to us, which is important, which is what I think we are all, I think pretty much the TB community or the people working on this technology and others are focusing on, right? It's great, okay, there's data, all of that, but to actually make an impact, we need to be out there and get countries to get the uptake and so on. So I think one big one is these implementation pilots that some countries have taken on. I think we're generating some really good evidence there in terms of the operational feasibility. So I think, again, performance is not something we need to keep that's there. It's a molecular test. If a mutation is there, it'll find it. That's okay. But what does it mean in terms of getting actually samples there? This is, in a way, you're adding a technology to an existing workflow, right, to an existing sample workflow. So I think part of it is understanding where this will sit in the country. So some of these implementation pilots would actually give some of that data that I think can be utilized for countries with very similar archetypes. We can't expect to do pilots in every country, but I think whichever countries have done this and done this successfully, beyond the countries we're working with, Bangladesh, Eswatini, Kyrgyzstan, or others. So with Stop TB and other partners have worked here, but they've really shown that yes, it works, but also in our setting, this is where we want to utilize it. So I think doing it in a smaller scale, say in area or one province to understand how the samples will come in, what does it mean, how quickly can it actually be a full sequencer? Do we run a single bag, multiple, all that? So all those operational aspects, that's one I think will be a real benefit, and actually for us to document all that so that people have a blueprint, if you will, to actually utilize. The second one, of course, is the funding aspect of it. I think that's going to be, I think, ensuring, I think Global Fund now has that, but making sure all these products itself are on these Global Fund and GDF procurement lists. And that is something countries are made aware of and they are able to procure. And also with access pricing, obviously it needs to be something that, knowing that the volumes are there, countries actually, or companies understand there is, from their side, it's still a huge market, but from the perspective of there is the big unmet need that exists. So I think that access pricing and getting on the list. I think the third one I'll conclude with, it would be this and this and this. Sorry, that's the third one is always the hard one. So I think the training aspect was one I wanted to say, but I think that is, again, that exists. That's something that's standard, that I think in a way the TB community has done that for different diagnostics. But I think one of the things here is to actually, because we're taking advantage of a technology that can be used for every disease, it is also that thing of, hey, that it can actually be shared. So it should not, all the burden and the cost should not be just on TB, but it is something that can be. So it's really with ministries of health and really the voice of advocacy to understand how that can be. And ministries can actually make those sound decisions to say, you can get the most out of that investment that sits there. So that's a little bit more nuanced to understand what that means. So things like calculators or costing tools to say the forecast, budget, what this means. If you add 3 diseases to this, that means you're going to literally pay off. There's a return you're going to see within a few years itself and so on. So that would be your third aspect that I'm highlighting.
Thanks for sharing that. I think funding, access, how to convince people to use the existing infrastructure for something that's close to us is always a challenge. But on that note, I had a very interesting question come in from one of the members in the audience, which is, what would be the timeline to include newer drugs like BDQ and PTO resistance in target NGS testing, since we are more concerned about TB? So
for bedaquiline right now is already included in the current tests on the market. It is at about, I think one of the tests at least, it's about over 80% sensitivity compared to culture. So 80% is still better than 0, which we have if we didn't have any other tests. So I think that's good thing is that is included. To include the other drugs where I think it essentially is like how, in a way, we don't want resistance to emerge too quickly. So you don't want to be seeing resistance predominate right there too soon. As far away as possible, the better. But unfortunately, that's the data you need to make that link between that mutations. Okay, we know there's resistance, clinical resistance or phenotypic resistance. But if we sequence, these are the target, these are the mutations or changes, and that's what then I can capture in my test. So in terms of going back and making the change, if it's just a bioinformatics change, I mean, in terms of how long it takes, that time frame then becomes a matter of a few months, really, because they can make the change, we'll do a reanalysis and so on. But if it means they have to add an entire new gene, that might take a little bit more time, but it's still just adding an additional primer and for that primers for that targeting step. So in a sense to say it is still within shorter than it would take to develop a brand new test once you have that information. But to get that information in the field for new drugs, we hope it won't be too soon because you also don't want to see, but where there is resistance emerging, we hope that information is captured and can be actually fed back to be able to design and update these tests. I hope that's clarifies.
Yep. Always, like we said, right, like protect the drugs, but at the end of the day, we need to do that to make sure that patients are okay. So this, again, super promising. And on that note, another question is, what could be the best approach to advocate for targeted NGS?
So this one, I know Ramya would probably be covering a lot of this. So I'll leave it to the experts to answer this. But I think from our side, I think absolutely. I think first making sure we answer all the questions that the community has, right? So I think this is sort of hopefully just a teaser, but there's a lot more. We can spend hours probably, inundating you with material, but it's really about what do you see as the main big things, right? If you've seen this already, you've seen previous diagnostic, you've seen things that failed, things that took too long. It shouldn't take that long when you have something that's working and out there. So those learnings, hopefully that's something you guys have seen as a community and the CCSE groups. to be able to answer that. So I think having some way to understand what are those things? Is the barriers just about the knowledge of it, understanding it, or is it a bit more to be able to talk to governments, kind of give them that place? So it's not just the Ministry of Health, because in most parts, ministries of health seem to get the value of this, or the labs definitely get the value of this. But it could be that Ministry of Finance and sort of that overall thing across different diseases, right? That's sinking. And those are always a bit more complex, but that's where I think groups like you guys can actually make that difference. There's only so much the technical people can do, but we really rely on that to get that word out. But really, what are the gaps you see or things that you have? No question is too basic to me. In fact, your questions would be the most difficult to answer, but those would be the most important we need to answer for you to be able to do the best to get countries to actually uptake this, so everyone gets benefit from this.
Makes sense. And I'm guessing we'll hear a lot more, like you said, on that front later. But one last question before I quit on this one is, what's next? So I think in your presentation, you mentioned that there's a WHO policy recommendation for targeted NGS, but what's next? Where do we go from here?
Yeah, so I think some of the things we just covered, those would be ones, right? So implementation, right? So understanding from countries, but actually completing that, generating that, in a way, the operational guidance beyond just saying, okay, this is clinical trial, this is all controlled setting. That's not what you're expecting. What is it in a real life setting? What are all those within the messy context of all of the things that are happening in a TB lab? Does this work? Like, what are the things, challenges? What are those bottlenecks, and what needs to be fixed? Is it a technical fix? Is it a operational fix? Is it a manpower fix? That aspect. I think the next one is, of course, as we say, it's not yet at a as high a sensitivity as you want for the new drugs. Of course, a lot of the knowledge doesn't exist. We know the knowledge, as knowledge improves, the sensitivity performance is improving. So I think in that case, it's about potentially WHO looking at updated evidence. Hopefully, I think they arrange, typically for new diagnostics, they have the guidelines development group meeting, and then subsequently potentially technical advisory group meetings to look at new data so that they can update the policy. So that in a way gives that confidence to countries and end users, saying, okay, actually the performance is good enough. Now I can think of rolling this out or scaling it up. So I think that would be a second big one. And these are things within the next 1 or 2 years that we are talking about to be key big ones that I'm really hopeful about.
Makes sense. Thank you so much again. I know we're running against time. There are a few more questions. We'll make sure we get to that through chat. But thank you so much, Anita. This was extremely helpful, and thank you for being patient and answering the questions. Back to you, Wayne.
Thank you, Saurabh. Thank you, Anita. So we're going to move to the next presentation, which is on tNGS advocacy, priorities, and actions. Our presenter today is Ramya Anantha Krishnan. She is the director of REACH and a core group member of the Stop TB Partnership's New Diagnostics Working Group. So Ramya, the floor is yours.
Thank you very much, Wayne, and a very good morning, good afternoon, and good evening to all. Please let me know if you are able to see my PowerPoint.
We see it. It's not in presentation mode just yet. Now we see it. Go ahead.
So thank you for this opportunity, and I think it was interesting to hear the previous speakers, Anita and Saurav, sharing, Anita, the overview about the new generation sequencing, and also Saurav sharing how communities need to understand that. And I just want to start off with by narrating a small incident that coincidentally happened just a couple of hours ago. So I was meeting a group of TB survivors and TB champions, and there was a photographer there, and he was watching this program, and he came and told, "Oh, I'm just hearing about how TB champions can play an important role." And he was an elderly gentleman, about 70, 75 years old, and he said, "I also had TB." About 50, 55 years ago. And at that time, I had RC-Nex, I had INH, I had rifampicin, he was saying that. And so, how is it now? And it's been like nearly 60 years, and I'm sure you must have come a long way. We hear TB being a priority. They took my sputum sample and they looked into the microscope and identified the TB. And I think that was when, it really struck as to how much we still rely on those drugs. And of course, with the drug-resistant TB being a major problem now, we still have not come a long way. We do, as Aluchika mentioned previously, we do have some new drugs coming in, but we still also rely very much on our good old drugs, INH, rifampicin, ethambutol, pyrazinamide. And I think that is where this whole NGS is going to be playing a very important role, because with the new drugs coming in, how do we protect these drugs and how do we make this treatment truly meaningful to the people with TB? And I think that is what the advocacy priorities and action needs to be centered on. In fact, and again, I just want to narrate another conversation which I had with another person, and I was just sharing that we are having this webinar, and and I mean, they were surprised. Oh, so do you mean to say, I mean, the question the person asked me was, do you mean to say that we could actually know what drugs we were resistant to? And because this particular person had XDR TB, had undergone, and that too many years ago. So they had taken the first regimen, the 6 months regimen, then the 9 months regimen, and then again tested, and DST happened. And this, when I was talking to them about the next generation sequencing, that seemed like such a game changer. And for that, a person who had TB, and it was very humbling to see. I mean, this would really revolutionize a TB diagnosis and treatment. And I mean, literally, he was very emotional just hearing that something like this is in the pipeline. And this is what it means to the communities, to people who've had TB, and to the people who are probably going to still be having TB, at least for the next couple of years. The next generation sequencing is definitely going to be a game changer. And of course, I think the stage was set very nicely by Anita and Saurabh as to why we need to advocate for NGS. Of course, one is identifying the TB cases, but also being truly person-centric, because NGS is going to help identify the drug resistance strain, and it helps us to understand the transmission patterns within the community and within the family. Again, the time to diagnosis, that is going to be reduced, and as I mentioned in my conversation earlier, a person with TB need not go through several rounds of treatment before they actually know that they have a drug-resistant TB. And even now, with the advent of CBNAT, and even with the access to CBNAT, we have multiple and repeated sample movement, where the samples, we need to first send it for CBNAT testing, and then we send it for first line LPA, we send it for second line LPA, we send it for liquid culture. And though in a diagnostic sequence, in a cascade, this seems very clean on paper. But in practice, in many countries, what we are seeing is that there is not such a seamless flow of the sputum sample, because for every sample, the person has to come back again and give the sputum again and again and again. So I think that is one of the things that is a huge, of huge importance, is not only reducing the time to diagnosis and treatment, but also improving the quality of life. What is the quality of life of a person with TB if they have to keep repeatedly producing sputum samples every week or every month? Again, NGS is a huge game changer, as was mentioned in surveillance, and especially in settings where there is a huge movement of people, even within the country or country to country. There's a lot of migration, and it plays. It's very important to understand the strains that we are looking at, and whether they are drug-resistant strains or whether they are just innocuous strains. So I think, again, that is very critical. And of course, it overall helps in diagnosing people with TB who remain undiagnosed. Excuse me. So in terms of what the advocacy priorities are, I think for anybody to advocate, the knowledge is very critical. I think we need to know, to act, and to advocate. And I think a key, and that is where we really need to advocate now immediately, is how do we create the sense of awareness among our key stakeholders to create that critical pool of people in the community, in the scientific field, in the medical fraternity, and even in the policy makers who understand what the targeted next generation sequencing is all about. Because only with knowledge comes the action, and with action, we could definitely advocate. So I think it's one of the very key advocacy priorities is how do we create this awareness about NGS among our communities? How do we, it's very important to share testimonials of people with TB on the need for point of care testing. And how do we create this social media and grassroots level advocacy? And I think while we saw in the last 3-4 years, again, as mentioned by the previous speakers, with the huge investments that was made in the targeted next generation sequencing for COVID. So we have a huge pool of resources, the infrastructure, and in many places, even the healthcare and the system is geared up to do this. So why not leverage that? And again, that's a very clear advocacy priority. So how do we leverage those, that the investments made during the COVID times for TB, so that first it doesn't go to a waste, because as was mentioned today, the targeted next generation sequencing is by no means cheap. But at the same time, how do we utilize the huge investment that's already been made, and how do we use the data and the evidence that comes out of it to support the advocacy efforts? And I think, again, I would like to say that the advocacy priority is in the communities. Through the affected communities, we also need to reach out to the larger communities, because unless we create a demand in the community, we cannot advocate for bringing these newer technologies into the market and buy the program. Again, we also need to advocate in terms of evidence. We have many pilots which was shown, and we need to move from project pilots to programmatic operational rollout, and again, generate a lot of evidence. We need to collaborate with the policymakers and government agencies and identify these champions among even the policymakers, even among the private sector, among the clinicians, who would also understand the value of NGS and help their community also to understand the importance and use of NGS. So definitely, as I mentioned, we need research, we need evidence, and we need people with the technical expertise to be able to have the intermediaries who can then communicate the knowledge on NGS according to the different groups of people. I mean, how can we simplify it? How can we convert it into local language, and how do we create this interest, and how do we create the community demand? In all, I mean, just I would like to go to my last slide because of time. I think we really need a call to action for NGS implementation. A rollout because it really helps, and this is where very important to invest in NGS. We need to address the barriers. NGS gives us a platform to address barriers, improve acceptability, and NGS also helps us to ensure equitable access, and it also helps us to understand the transmission dynamics at both the community level and the household level. And we need, and it's our responsibility to encourage our communities, our affected communities, our multiple partners to understand more about NGS at every platform that is available, so that this call to action for NGS can be realized. So thank you very much for this opportunity, and over to Wayne.
Right. Thank you so much, Ramya. This was really good, and thank you. And you had a lot of the points that's why I knew you'd be best placed to answer. There's some of the things you're trying, but really learning from you guys in this one. So for the Q&A session, this next one, our moderator is Goodman Makanda from TV Proof. Goodman, the floor is yours. Please also introduce yourself. Thanks.
Thank you very much, Rumai, and my name is Goodman Makanda. I'm based in South Africa. I'm a TB survivor working for TB Proof as TB research and peer supporting the patient. As you mentioned, that is a nice presentation and straightforward. What is the plan, and how can we make sure we're mobilizing more when we talk about advocacy? But I don't think we do have a question, but I do have one. What are the opportunities to engage the civil society and the community as a champion to help the countries leverage their expanding diagnostic and capacity establishing during the COVID pandemic?
Sorry, I couldn't. Can you just repeat it, please?
Can you, what is the opportunity to engage a civil society and the communities to help the countries to leverage their expanded diagnostic capacity established during the COVID-19 pandemic?
I think what we see, especially in India and in many of our interaction in other countries also, is this huge people's movement. And when I say people's movement, it's a movement of civil societies, of affected communities. And I think we are heading to a critical mass, where there is actually a lot of noise being created on TB. And the knowledge on the diagnostics, on treatment, on access to treatment is all getting really amplified. And I think that is where this whole, I feel the entire civil society space, the environment is truly set up to understand the NGS and to take this forward. But in terms of what the lessons learned from COVID, they may, I'm not sure those nuances would be understood because of course, all of us knew about the PCR, and that was very clear. So then how do we relate PCR and the NGS, and how do we make the communities and civil society understand the similarities and the differences, because that basic knowledge is there. And today, I feel the larger communities understands that well. So we have a fantastic platform to build on. And I think this is a very opportune time to build on that platform laid by COVID, of course, unfortunately, but the platform is there. And I think we really need to strike when the iron is hot.
Okay, thank you very much. I like it because I think we did advocate more when it comes to COVID to say we do want a vaccine that will protect us. So things like this that now even the first presenter was talking about the new regimen and the testing tool that it should be available. So we need, as you mentioned on your slides, collaboration with the stakeholders, collaboration with the TB survivors. I think the voice without TB survivors in this innovation around this tuberculosis, it won't make more impact. We need to combat together to make sure this vehicle is moving. So I think the last question I will be asking you, because I don't see any question on the box, anyone can write a question. And if you don't speak English, you can write your language, and I think it will be translating to English, so everyone we can read the message and ask the question. This is the opportunity. This is the way of we're moving forward with when it comes to advocacy in the community, using our healthcare facilities, using our government itself, Department of Health, to make sure they are hear our voice, because without our voice in the community or in the department, we won't be heard. No change will be happening. No, we won't be talking about betaqueline. We won't be talking about the new regimen because we want to test and treat what should be happening. So are there any example of innovation advocacy in the countries that they are having a result in improving assess the rollout of the new TB technology that we should be thinking about?
So I think the way we are now, and I'm sure you and Saurav would agree with me, is that I think the presence of affected communities and civil society has been quite limited. And I think only in the last couple of years, it's building momentum. And in fact, the NDWG now has a separate group, the community engagement group, in the last 1.5 years. And so we see this engagement of the communities happening in different forums and different places. And I think what is important is how do we foster this dialogue between the technical people and the community? And that needs to happen at every step of way. Because if we expect the communities to advocate, set priorities, how do we take the technical knowledge that is coming and which gets updated, or which is the new things that come out? How do we take that information to the communities and civil societies in a periodic, sustained manner? What is the forum to do that? We need to have that forum where there is a continuous flow of information from the technical side, be it drugs, be it diagnostics, be it vaccines, and that continuous flow of information is needed from the technical group of people to the community people, so that the community have the bandwidth, they have the voice, but they need the knowledge. And I think that is where we need to sort of focus on. And again, I mean, not specifically for this, but of course, we see that there are many examples of, especially in-country advocacy that is taking place for reducing price of cartridges, or the price of bedaquiline, making BPAL regimens accessible. So we have countless examples, especially in the last few years, and I think that is where we need to invest more and more in engaging with the communities, building their capacity, and giving them opportunities and platforms to also share their advocacy priorities. Thank you.
Okay, thank you very much. I think the last question that is coming on the chat, after I think we will be leaving the stage. What is the cost of tNGS for the TB patient in developing countries? From Nautina, from Benin.
So I'm not very sure because it's not available. I mean, it's all being done by the program in a project setting. So it's not like available, but I'm sure it's thousands of rupees or hundreds of dollars. But I could definitely find and pass on that information.
Okay, thank you very much for your time and a nice presentation. We're looking forward. I should be asking what is the next step in coming for advocacy, but the panel is on back. Thank you very much for your time. Bye.
Thank you, good. And thank you, Ramya. So we're going to move to our final presentation by Austin Obifuna, the Afro Global Alliance Executive Director and the Stop TB Board Vice Chair. So Austin, the floor is yours.
Thank you, Anita. I hope you can hear me.
Yes.
Thank you very much. I'm looking at the time. I think we're already behind time, but I promise I'm going to be very short in terms of my presentation because it's actually straightforward. And this document you are looking at, I mean, the key TB ask for the EMA is a document that I believe that most of us in the TB community have seen and also have digested in a way. But coming from the lots of good presentations that have happened, before mine now, I think it's very important to also just put a word in terms of how important this what is happening now is, because often at times people are engaged either at the middle or at the last bit of every good initiative, probably maybe during implementation or during monitoring. But it's good that civil society communities are also involved as part of the planning, because even the beneficiaries as well could actually bring very important points to the table. And even to the fact of demand creation, because you may have the best equipment, you may have the best facility, but without creating demand, good demand for it, and also obvious reasons, because these are in the communities, it may be very difficult to assess such. So this is really, really good. And as a result of, is a new tool. Sequencing is something that most people see it as science, and within the civil society and the CSOs, it is always very good to be able to unpack some of these initiatives so that they can actually know what should be their contribution, how to support the initiative, and also do strong advocacy, and also call for stronger political will. Because at the end of the day, if it's being piloted, we're also looking at scaling up most of this intervention. And the message also came up this morning in terms of sequencing also helps to avoid unnecessary extensively drug-resistant TB, because it's a kind of preventing this from happening, because understanding what you are going to treat, understanding what the situation of that patient, because it's person-centered, looking at the individual. So it also creates a better opportunity for us to have good results and also value people's life, and not just playing politics with people's life. So let me start on that note, because I think mine is very, very straightforward. And before I even go on, I just wanted to say that I shared a link, a petition that we all have on financing, because even if we're talking about sequencing, if we don't have money, if we don't have finances to be able to promote such good initiatives, I'm sure that it will be the same cookbook approach, where we have all these good initiatives, but it will never see the day of life. So it's very important that as we speak to this, we also promote channels on how we can be able to move forward with the sequencing initiative. So first of all, let me say thank you to Stop TB Partnership for helping to put together this, helping the entire TB community as a whole, to lead the process of putting together this key TB ask, because it's a product of the entire TB community. We had a lot of inputs from the entire TB community, from the whole world actually, on this, to make sure that we have a clear roadmap on how to engage in the space of AMA. And I want to say that the AMA is actually a synonym to drug-resistant TB. You cannot talk about AMA without talking about drug-resistant TB. I mean, that is the overall principle around it. So we must, at every time, put TB in the at the front space of AMA, because that is where it belongs in a way, as one of areas to focus on greater interventions around TB. And it brings us to, please, next slide. It now brings us to next again. It now brings us to the key acts that I want to speak about, that we also will be discussing in New York this coming week. We already had a chance of engaging with the facilitators, engaging with a broader multi-stakeholder group in May to share these key acts. I must say that, yes, some of these key acts were taken into consideration, but not in full as we may want it. But I can say that there are a lot opportunities for improvement, and also to do more justice in terms of adding, most of these asks, especially around political commitment, around accountability as well. But most important is the financing. So if you talk about political commitment around AMA, I think it is very key. It is very important, like I said, that TB, you cannot speak about AMA without bringing in drug-resistant TB. So when it comes to political commitment, we need very strong indicators, and not just strong indicators, but a tracer indicator that puts TB in front, the global and even the national AMA strategies. Because if you look at the progress, on TB and the TB R&D, it's critical, indicators of the progress on AMA. And this is really, really, really important for us to be able to do that. Then it brings us to financing. Next slide, please. It brings us to financing, which is another very strong, somebody will tell you that, oh, it's not about money, but without resources, without funding, you will not be able to do much. Countries are struggling. And that is the essence why we need to be able to push, very strongly to have diversified, probably diversified source of funding for TB, because it looks like the global fund is shrinking, is becoming a little bit more just a one source of funding for TB, not just a major source of funding for TB, because and also creating a lot of confusion around the epidemic, the TB epidemic, because we need to really work to speak, to walk the talk, and not only just speaking about TB. TB, we know today, is #1 infectious killer, if in the world, and also have shown that, put together most of the global fund diseases, that TB is actually the highest number, in terms of mortality. So, and even killing people with HIV as well. So we need to really, really, do more in terms of commitment, in terms of resources for TB. And now we are talking about drug-resistant TB. We heard when Luchike mentioned about drug-resistant TB. We need even to do more, because we used to call it a time bomb, but I think it's already exploded, and all we need to know is how to catch up and be able to roll it back. And it's becoming increasingly very important that we speak more, around the drug-resistant TB and the financing for it. Next slide, please. We'll talk about diagnostics, we'll talk about treatment, we'll talk about prevention. And like I said initially, that also this sequencing is also a way of preventing, multiple resistance that can happen, if not knowing the person. And you see, I like the fact that the patient-centered approach has always been promoted at the UN. And this is also a way of also making sure that we have a person-centered approach to the TB response around the world, because sequencing will always, kind of give us a prior understanding about what the mutations the patient may have. And this is really, really good. And also now it takes, another one, which is very, very key, is on community rights and gender. Please, next slide. Is on community rights and gender. Yes, I talked about engaging people, but not just engaging civil society. And I mean, engaging them in the right way, engaging them with evidence, engaging them with countries, understanding what their epidemic is like, understanding who are their key and vulnerable population, understanding people who need to be targeted, giving special differentiated targets. And this is why we're even promoting the patient-centered approach. And this is very key. And we need to really, really, really promote. I can say that thanks to Stop TB Partnership, because apart from the challenge facility for civil society, yeah, it can hardly, tell me, there is no other mechanism that actually trickles real resources to the lowest level, to the periphery, to the communities at that level, to promote their activities, to promote their CRG assessment, which is being done in the countries and showing evidence, to promote even the one impact approach, because this is another accountability mechanism that I'm going to speak about later when I speak about accountability. But this is an approach that if we support civil society, to extend that services to the unreached, to extend that services to the vulnerable population, to prevent stigma and discrimination that actually bars, creates a huge gap in terms of services and also assessing services. So this will be one area that I always want to push for, community rights and gender, and pushing for it. I'll be finishing soon. Next slide talks about research and development. And we talk about prevention, we talk about vaccine. Every day we are talking about vaccine in TB. This is, since I joined TB response, we've all been talking about vaccine. And vaccine that we have in TB have been about 100 years. I always give this example about COVID, the political commitment, the political will. COVID is a disease that was never known by anybody. Within less than 2 years, we were able to have a vaccine for COVID. And we're still struggling, on how to develop vaccine for TB, which is also a very good. That would be a game changer, because it's also part of prevention as well. We need to really push, and if there should be some resources in AMA, it should be focusing on also how to promote vaccine development in TB. And finally, next slide, we're also talking about the health systems and accountability, like I mentioned, on the one impact approach. We need to really have, people outside that chain of command doing the monitoring. Even if it is an independent panel on evidence for action against AMA, that should, because you cannot be a judge in your own court. Because for me, I see it wrongly for the fact that you should allow also the public sector to do the monitoring and evaluation of these activities. You can do it to be able to monitor your program, to make sure that you're doing well, also you're hitting the target. But we need people of independent minds, especially coming from the civil society as well, to look into the program and also give a critical analysis of how to make things better, in terms of these initiatives. So I will want to quickly stop here and then go on to closing the section, because I know that we are already behind time. But it's very important that these key asks are being pushed. I know a lot in the coming week. We may not be doing a lot of our usual mission, but it's also very good that if you have a opportunity of engaging with any of the mission, please do that. Getting feedback, from the TB committee. And also we know we also behind already. We have few people, TB, and we need to really recruit people to be able to do that. I want to stop by saying that I have this message from Chika, which always resonates with me. He says that we want people and the wider AMR community to own the fact that drug-resistant TB is an important component of the AMR and the leading cause of death among antimicrobial-resistant infections. With UN high-level meeting on AMR being discussed this year, the community cannot afford to stay quiet or silent, which is very, very important, not just only on this AMR, but also on Global Fund. And I am really pleased and excited that we came together to develop the key ask for UNHLM on AMR. And this key ask is very comprehensive. I wouldn't say a perfect document, but if there's a word like perfect, I think it should be called a perfect document. And it's a living document we can always add, we can always remove, but the most important thing is that as we move forward, we develop a lot of things will be happening in future, and we should be able to have these things added to the key ask. I want to stop here and just to say that first of all, thank you everyone for joining this call. Well, I think I can go ahead and close because I'm not sure there will be an opportunity for any question because of the time.
Yeah, thank you so much, Chief Austin, for this presentation on the community asks. We are well over the end of the webinar time. Unfortunately, there are still so many people online, an indication of how interested people are. So we are going to be collecting the questions that are in the Q&A box. We'll be working with the presenters today to answer them all written, and we'll share the answers by email to everyone who registered for the webinar. You'll also get the copies of the presentations and links to the recording. So I know we're over time. We do want to ask a few very short questions to all of you about today's webinar. There should be a poll popping up. So before you sign off, we'd really appreciate if you could just take a moment to answer them. And on behalf of us at Stop TB and FIND, we'd like to thank all of the speakers and all of you for attending. So we'll be in touch as soon as we have the dates ready for the next webinar in this series. And again, thanks and goodbye.
Thank you.